How you doing, everyone? Welcome to this lecture on for your module for exercise physiology. Today we are going to start our discussion about the third energy system. We're going to talk about aerobic metabolism, or the aerobic production of ATP. Today we're going to focus on the aerobic production of ATP from carbohydrate. There is a separate process for the aerobic production of ATP from fat, which we'll cover in the next lecture. So everything we're focusing on here starts with carbohydrate. Before we move on to that, let's have a very, very quick recap on what we've covered in our previous two lectures on the energy systems. So we started out looking at energy system number 1, the ATP PCR system. This is an anaerobic system. It doesn't require oxygen for it to occur. It's very, very fast. It provides almost instantaneous production of ATP when we require it. However, it has a very limited capacity. It will only provide us with enough ATP to last for about 8 to 12 seconds of activity, depending on the intensity of the activity. The more intense, the higher power output, the less time we will have, energy we'll have from this system. It uses phosphocreatine. That's the PCR, phosphocreatine, as its energy source. That's the fuel to phosphorylate ATP from ADP. So when we say we want to phosphorylate something, we are adding a phosphate group on. So ATP, adenosine triphosphate, is broken down to ADP, adenosine diphosphate. That releases energy, which we can use to fuel activity, and it frees up a phosphate group. We then want to reattach a phosphate group onto the ADP to recreate or replenish our ATP stores. The ATP PCR system uses phosphocreatine to do that. So by breaking down or oxidizing the phosphocreatine, it releases a phosphate group and a little bit of energy. That little bit of energy is like the superglue that reattaches the ADP, the phosphate to the ADP, replenishing our ATP stores. Okay, so that's a very quick recap of that system. We then went on to discuss glycolysis. Now, glycolysis is a slightly more complicated system. However, there's some commonality. It's also anaerobic. It's fast, but it's not as fast. So it can provide us with a fairly quick supply of ATP. Not as fast as the ATP PCR system, but it is still pretty quick. It has a better capacity, so it can provide us with enough fuel for activities lasting for minutes rather than seconds. So maybe up to 2 to 3 minutes of high intensity activity can be fueled by glycolysis before we will start to run out of energy or fatigue will kick in. Glycolysis uses either glucose or glycogen to phosphorylate the ATP from ADP. So glucose or glycogen are both forms of carbohydrate. Glycogen is stored in the muscle, glucose is in the blood. It will use either of those as its energy source, and it will break it down in order to provide the pieces of the bits it needs to phosphorylate ATP from ADP. This system produces NADH and pyruvate or lactate, and that's important because that's where we're going to pick up the discussion now when we talk about the aerobic production of ATP. So that's just obviously a very quick recap. You need to be familiar with those other two energy systems, so make sure you're fully familiar with what's in the previous lectures before you move on from this point. If anything's not ringing a bell there, go back and recap it before you move on. Okay, the aerobic production of ATP from carbohydrate, I should add in at the top of that slide there. This is the third system of energy production, and it's the oxidative system. It's the most complex of the three energy systems, and we're only going to provide a fairly brief overview of the key points here in this module. It'll be enough for you to understand the broad strokes of aerobic metabolism, of how carbohydrate is broken down, but it is very, very detailed, and I'm not going to do a deep dive on it. We're going to give you just the broad strokes, enough to understand it. So one of the key points here is that this is an aerobic process. Oxygen is required. Okay, we'll see why in a little bit. This system is also sometimes known as as oxidative phosphorylation, because we are phosphorylating ATP from ADP in the presence of oxygen. So oxidative phosphorylation. Previously, the two systems, as mentioned, have been do not require oxygen for them to work, so they can be termed anaerobic. The anaerobic production of ATP from the ATP PCR system and glycolysis occurs in the sarcoplasm of the cell. The oxidative production of ATP occurs within special organelles called the mitochondria. Everyone should have heard of the mitochondria, the powerhouse of the cell. I don't think you can mention mitochondria without saying that. In the muscles, the mitochondria are found adjacent to the myofibrils, and they're also scattered throughout the sarcoplasm. Okay, one of the adaptations to exercise training is that you can increase the number of mitochondria that are in your muscles, and if you increase the number of mitochondria that are there, you increase your capacity for aerobic metabolism to occur. So more mitochondria will allow you to produce more energy. So we've seen this slide before. This is where we finished up with the summary slide when we were talking about glycolysis. Muscles need a steady supply of energy to continuously produce the forces needed to produce long-term activity or endurance activities, I should say. So unlike ATP production, the oxidative system is quite slow to turn on. So the ATP PCR system turns on really quick. Glycolysis turns on quite quickly. However, the oxidative system is fairly slow to turn on. But once it is up and running, it has a much larger energy producing capacity than either of the other two systems. So, for this reason, aerobic metabolism is the primary method of energy production during endurance exercise. So these are any activities that last longer than a few minutes and up to multiple hours in duration. Okay, so technically anything beyond, 2 to 3 minutes in duration is considered an endurance event, an endurance activity, because aerobic metabolism will become the primary source of energy production in that activity. This places considerable demands on the cardiorespiratory system, so your heart, blood vessels and your lungs, because we need to deliver enough oxygen to the active muscles in order for this aerobic metabolism to take place. The oxidative energy production here comes from carbohydrates. That's what we're going to start to focus on. And this starts with glycolysis. So the oxidative metabolism of carbohydrates begins with glycolysis. We can also aerobically metabolize fats, but as I said, we'll talk about that in a different lecture. And protein contributes very, very minimally to this in exercise conditions. So we're not really going to discuss the metabolism of protein here. So as I've said numerous times now, we're going to focus on the oxidation of carbohydrates. So glycolysis plays a role in both the anaerobic and the aerobic production of ATP from carbohydrates. The end product of glycolysis is the production of 2 pyruvate molecules. So we see those here. Glycolysis, we know that it's a 10-step system, and it gives us 2 or 3 ATP molecules depending on what the original source of carbohydrate was, and it also gives us 2 pyruvate molecules. And we said that those 2 pyruvate molecules can then go on to be metabolized aerobically to produce more energy, and that's what we're going to look at today. So specifically what happens is, in the presence of oxygen, so when oxygen is there, the pyruvate produced from glycolysis can enter into the mitochondria from the sarcoplasm. So glycolysis is occurring in the sarcoplasm. Aerobic metabolism of the oxidative system occurs inside the mitochondria. So those pyruvate molecules can enter the mitochondria where they undergo further oxidation. There's 2 cooperating metabolic pathways at work here, and they're both required to complete carbohydrate metabolism aerobically. So if we want to fully break down carbohydrates aerobically, we need these additional 2 systems. They are the Krebs cycle and the electron transport chain, and we're going to look at both of those in a little bit of detail now over the next couple of slides. So the Krebs cycle completes the oxidation of pyruvate, which is formed from glycolysis. It involves a complex series of enzymatically catalyzed reactions that you can see illustrated in the image there. Don't worry, you don't have to memorize what's going on there, but we do want to understand briefly what's going on. In the presence of oxygen, the pyruvate generated via glycolysis passes into the mitochondria from the sarcoplasm. As it does, it's converted into a new compound called acetyl coenzyme A. That's just often written down as acetyl CoA, and we can see that at the top of the Krebs cycle here. As part of the Krebs cycle, the acetyl coenzyme A binds with a molecule called oxoacetylase, which is here, okay? So we have the oxoacetylase, it binds with the acetyl coenzyme A that's entering into the mitochondria, and this creates citrate, okay? Citrate then continues on through the Krebs cycle. I'm not going to go through each step of the Krebs cycle because, as I said, it's quite complicated. It's more complicated even than is illustrated in the diagram there. However, what we need to know is that each molecule of pyruvate that enters into the Krebs cycle is converted to acetyl coenzyme A, binds with oxoacetylase, and produces citrate. Citrate will then go through one full turn of the Krebs cycle. Okay, so this citrate here passes through all these steps and back up to the top of it. As it passes through, it produces one molecule of ATP, it produces 3 molecules of NADH, and it produces 1 molecule of FADH. So remember, we said at the end of glycolysis there's 2 molecules of pyruvate produced, and they both enter the Krebs cycle. So the diagram that we see here and the diagram that you see in all the books only will ever illustrate one turn of the Krebs cycle, but everything happens twice because both pyruvate molecules are oxidized. Both pyruvate molecules produced via glycolysis will enter into the mitochondria and follow through the Krebs cycle. It's called a cycle, the Krebs cycle, because the starting molecule, oxoacetylase here, is regenerated at the end of the cycle. So it's self-perpetuating. So the acetyl coenzyme A, the pyruvate enters the mitochondria and is converted to acetyl coenzyme A. It bonds with the oxoacetylase to produce citrate. Citrate passes through the cycle and ends up as oxoacetylate again, ready to receive more acetyl coenzyme A via pyruvate. So it's a cycle that can continue provided there's enough oxygen. So energy production so far, we've said we're focused that the ATP PCR system produces a limited amount of ATP. We're not going to focus on that. Glycolysis, we've said, produces either 2 or 3 molecules of ATP, depending on whether we use glucose or glycogen as the starting molecule. It also produces 2 molecules of NADH. Plus, from glycolysis, we have 2 more molecules of NADH are formed during the conversion of pyruvate to acetyl coenzyme A as it enters the mitochondria. So during that conversion, we get 2 more NADH. The Krebs cycle, 2 full cycles of it, gives us 2 molecules of ADP, 6 NADH, and 2 FADH. Now, we've mentioned these a lot, the NADH and FADH, but what exactly are they? We haven't actually discussed what they are. We've only mentioned them briefly to this point, but we see that they keep coming up over and over again. Well, to discuss NADH and FADH, we need to discuss NAD and FAD. So they stand for nicotinamide adenine dinucleotide, that's NAD, and flavin adenine dinucleotide, FAD. Again, don't worry, you don't need to remember those names. NAD and FAD are molecules that pick up hydrogen and some high energy electrons. And in the process of picking up that loose energy and electrons and the hydrogen, NAD and FAD are converted into NADH because they've had the hydrogen added onto them, and FADH because it's H squared because they've had 2 molecules of hydrogen added onto them. This occurs, so this mopping up or this picking up of hydrogen and energy occurs during glycolysis and the Krebs cycle. We've said that these molecules are produced during those cycles. basically NADH and FADH are high energy shuttles. Okay, so they pick up high energy electrons that are freed up during those processes, and they carry those electrons over to the electron transport chain, where they will power the continued production of ATP. Okay, so as part of glycolysis and as part of the Krebs cycle, as the various chemical processes take place, there's hydrogen released and there's high energy electrons released. The NAD and the FAD go around and they mop these up, they tidy them up and clear them up. In the process, they are converted to NADH and FADH, and they are used as high energy shuttles. You can literally think of them like the shuttle bus there in the picture. They pick up this stuff and they transport it over to the electron transport chain, where it's further used to produce ATP. Looking at it in a little bit more detail, why does this occur? During glycolysis and the Krebs cycle, if the hydrogen that produced was allowed to accumulate, there would be a decrease in the pH. If there's a decrease in the pH, it means the acidity is going up. Things are becoming, the environment is becoming more acidic. The environment is becoming more acidic. We've mentioned this in the previous lecture. If the environment in the sarcoplasm, if there's too much hydrogen accumulating, if the pH is decreasing and therefore the acidity level is increasing, this interferes with the ability of certain enzymes to function. The one we mentioned specifically was phosphofructokinase, which is one of the glycolytic enzymes that's important to the breakdown of carbohydrate. So if the hydrogen accumulates, the pH will drop, the acidity will go up, and that will result in the enzymes not being able to work properly, and that will limit glycolysis and stop you from being able to produce energy. It will fatigue you. So if there's too much hydrogen, it decreases the pH, which increases the acidity, which interferes with the ability of the enzymes to do their job, and it slows down the production of energy. NADH and FAD mop up this excess hydrogen in an attempt to prevent that decrease in pH and keep everything working normally. And as we said then, they're shuttled over to the electron transport chain, where they're used to generate more ATP. So if we look here, we can see step 1 up here, we have glycolysis, and we can see there's 2 NAD and 2 NADH produced. As we go into the electron transport chain, as the pyruvate enters the mitochondria, we have more produced. As we go through the citric acid cycle, we have more of these NADH and FADH molecules being produced along the way. They are then taken over to the electron transport chain. The electron transport chain, right, we've mentioned this a couple of times. Again, this is another fairly complex series of reactions that completes the oxidation of carbohydrate and the production of energy. We've said multiple times, glycolysis and the Krebs cycle results in the production of NADH, FADH, and some free hydrogen. They are all passed over to the electron transport chain. It's a complex series of steps, but basically the hydrogen and the high energy electrons that are carried over in those high energy shuttles are used to further phosphorylate ATP and produce ADP. This is known as oxidative phosphorylation. Okay. The result of the electron transport chain is the formation of ATP, that's what we want, energy, and also water, H2O. The H2O is formed by oxygen accepting the electrons that are passed down through the electron transport chain. So we see here, this is the electron transport chain, and we can see that we have the NADH, the FAD, and the loose hydrogens are all accepted in at various stages. They are broken down, releasing the electrons, which produces releasing the electrons and the hydrogen, all of which helps the process produce ATP. At step 3 here, we see that the end process requires oxygen. So oxygen is the final receptor for the electrons that are passed down through the chain. So this is why we say this is an aerobic system, because the very, very last step requires oxygen. If there's insufficient oxygen to accept the electrons that are being passed through the electron transport chain from the Krebs cycle that came from glycolysis, well, then the whole system backs up. Okay, so the delivery of oxygen to the mitochondria here is critical in order for these for this energy system to work. Without oxygen, the process would back up and stop, and that's why this system is considered an aerobic system. That's also why people with a high VO2 max are able to perform better, because they have a higher capacity to deliver oxygen to the working muscles, and because they've got a higher capacity to deliver oxygen to the working muscles, they've got a higher capacity to run things through the electron transport chain. Okay, so that hopefully that makes sense to you. The more oxygen you can deliver, the fitter a person is, the more oxygen they can extract from the environment, deliver to their working muscles, means that they can get this system working at a greater rate. Okay, they can go through the electron transport system more effectively. So the end product there is ATP and water. So exactly how much ATP is produced? So the energy yield, so how much energy we get from the oxidation of carbohydrate. We've got 3 steps going on here. First of all, we've got glycolysis at the top there. That's our first step in the oxidative process. We need that to occur. Then, oh, sorry, that shouldn't light up next. Oh, nor should that. We've got the Krebs cycle. This is what should have come up next there. Sorry. We've got glycolysis first. Then we've got the Krebs cycle, and then we've got the electron transport chain, which accepts some of the products of glycolysis and some of the products of the Krebs cycle there. Once all of these steps are complete, we can say that we have completed the oxidative metabolism of carbohydrate. And we can see here that the various steps produce various amounts of ATP. Specifically, sorry, I should have said this on the other slide. Every NADH molecule that passes through the electron transport chain will generate 2.5 molecules of ATP, and every FADH molecule that passes through the electron transport chain generates 1.5 molecules of ATP. Might sound odd that it's 2.5 and 1.5, but remember they always pass through in pairs, so we're actually getting 5 and 3 molecules of ATP. If we add it all up here along the way, I won't go through each one of those, what we see at the end here is the net energy production from the oxidation of 1 molecule of glucose is 32 molecules of ATP. So if we add all these up, so it's the ATP that is actually produced by glycolysis and a little bit by the Krebs cycle, but then it's the further oxidation of the NADH and the FADH that's produced in the electron transport chain that really gives us the bulk of our ATP production here. So 1 molecule of glucose will give us 32 molecules of ATP, and if we use glycogen as the original source of carbohydrate, we get 1 extra molecule, we get 33, because we didn't have to invest that 1 molecule of ATP at the start of glycolysis. So that is a brief overview of the oxidation of carbohydrate. Let's have a little summary of it there. So first of all, the breakdown of carbohydrate begins with glycolysis. This is an anaerobic process. The pyruvate produced from glycolysis can be further broken down aerobically. Each pyruvate molecule that's produced by the Krebs cycle and produced by glycolysis enters the Krebs cycle. This produces some ATP, and it also produces NADH and FADH. The NADH and FADH are high energy shuttles. They carry high energy electrons and hydrogen molecules over to the electron transport chain. Once they enter the electron transport chain, they produce more ATP. This process requires oxygen. So in order to pass the electrons through the electron transport chain, the very, very last step requires oxygen for the production of water, H2O. So without enough oxygen, this whole system backs up. The product, or the end yield, is 32 molecules of ATP if we use glucose, or 33 molecules of ATP if we use glycogen, plus some water. So we see at the end of glycolysis, we said that we get either 2 or 3 molecules of ATP. However, aerobic metabolism, the aerobic oxidation of carbohydrate, produces tenfold the amount of energy there. So it is much more efficient system. However, it requires a sufficient supply of oxygen for it to occur. So look, I know there's a lot there. Hopefully it all makes sense as we've gone through it. Please don't be afraid to stop, pause, go back, listen to what I've said, have a look at the notes on the PowerPoint presentations, including the notes that are underneath the slides. And as always, if you've got any questions, please direct them to either SJ or myself. Okay, thank you very much for your attention. That's it for today. I'll catch you in the next lecture.